closer together, we shrink the H zone. And as we pull this actin in that direction, we shorten this part over here, which is the I zone. So that should match up with what we saw in this figure. They're now combining the cross-bridge interaction with what's happening at the level of the sarcomere. Okay? All right. Okay, so now what we need to do is do this mechanism incorporating a phosphate group to get the change in the shape of that myosin head so that we get the interaction plus the power stroke. Okay, so I'm just going to change the focus a little bit. Again, you guys have these figures that you can have next to you while you're watching the video. This should just help you figure out which videos you need to be looking at. So let's start here at the top. Binding. When calcium levels are high, the energized cross bridge can bind to actin. Okay? And then it says ADP and an inorganic phosphate group are already bound to the cross bridge. So here, let's just take a look at one of these. Right? They're linking together two, but at this cross bridge, there's already an ADP plus an inorganic phosphate group sitting there. Okay? Now, this should tell us something already. In order to get an ADP and a phosphate group, chances are pretty good we started out with an ATP at some point in the past. And we hydrolyzed that ATP, and we transferred energy with that high-energy phosphate group, and the energy was transferred from the high-energy phosphate group to whatever the phosphate group was attached to, yielding a low-energy phosphate group as the energy was transferred. Well, the energy in the past was transferred into one of these heads, so this really represents a higher energy conformation. Okay, what we're looking at is the high energy conformation of this myosin head. This is where it has more energy. Okay? Now, the ADP and phosphate group that's inorganic, that's low energy, is already attached to the myosin head. Okay? Calcium is high. It binds to the troponin, which pulls the tropomyosin off of those binding sites. And this fits naturally in with one of these binding sites on the actin, right? So these heads are just going to stick right into it. As soon as those sites are exposed, those myosin heads stick into those actins. Okay? Now, the release of the inorganic phosphate group. We already transferred energy to the myosin head in the past. Now that inorganic phosphate group is falling off of the molecule, and when it falls off, it causes the cross bridge to do this. It kind of pulls this way. This is low energy, this is high energy. We bound the myosin head in a high energy conformation, and now it's going back to its low energy conformation. But as it moves to its low energy conformation, it's attached to the actin at these binding sites. So as it moves from high to low energy, it pulls the actin along with it. Make sense? And that causes the movement, right, the shrinking of that H zone in the I band. The power stroke moves the actin filament toward the H zone, and then the ADP is kicked off. So here, ADP and phosphate groups sitting there. Calcium removes the binding sites. Bam, we stick. We lose the inorganic phosphate. Power stroke, we lose the ADP. Okay? phosphate group kicked off with the binding. The power stroke is this. This results in the loss of the ADP. Now, to get this back off, right, to get it off of that actin, we have to get the myosin head back to its high energy conformation. So to do that, we're going to bind ATP to it, causing the cross bridge to detach from the actin filament. Okay? So ATP binds and pulls the myosin head back off of the actin to detach it. Okay? And then we're going to reset it so that it can do another power stroke. Resetting just means hydrolyze the ATP. So far we've got bind, release the phosphate group, pull in the power stroke, release the ADP. Attach ATP, pull it off, hydrolyze the ATP. That phosphate group provides the energy to get it back into the high energy conformation. And by it, I mean the myosin head. Okay? ADP, phosphate group attached, bind. Kick off the phosphate group. Power stroke, kick off the ADP. Bind the ATP, pull it off, hydrolyze the ATP, reset it into high energy conformation. Okay? That's what this figure is doing. Bind, power stroke, detach, reset. This is from a different textbook. I like it. It shows the ATP hydrolysis a little bit better than in this one. But the idea is this kind of binding, power stroke, detaching, and resetting is all about interacting phosphate groups, ADP and ATP, with the myosin head to alter its binding affinity for those actin sites. Okay? Again, one last time. The ADP and inorganic phosphate group are sitting on this molecule. The myosin is in a high energy conformation. The actin is there. Calcium comes in, we expose those binding sites. The myosin head sticks. The phosphate group and the ADP get pulled off. Right? We now move to the lower energy conformation of the myosin head. That pulls the actin, but it's stuck there. To get it off, you bind the myosin head with ATP, pull it off. That's the detaching step. And then we hydrolyze the ATP and transfer the energy to the myosin head to reset it into that high energy position. Okay? Bind, power stroke, detach, reset. Bind, power stroke, detach, reset. Bind, power stroke, detach, reset. Okay? All right, then I have some numbers here. I was just looking at that. You see my numbers here. All right, that thick filament, I made a mess of my notes. Good thing you can't see them. All right, each of the 350 heads of a thick filament. So if we look at a myosin molecule, that's roughly 350 of these heads, or 350 cross bridges, however you want to think about it. And each of these are forming and reforming these cross bridge with the actin, about 5 of these cross bridges per second. So when we do this, right, bind, power stroke, detach, reset, bind, power stroke, detach, reset, it's not like this, it's because each of these heads is forming 5 of these connections per second. So this process here is 5 times a second for each of these 350 heads that occur on the myosin thick filament itself. Okay? Does that make sense? this is what's causing the sliding in the sliding filament. As these bind and then they do their power stroke, that pulls the actin molecule in this direction. It slides the actin across the myosin. Okay? One last thing. We're doing this on both sides of the thick filament, so we're pulling the actin across the myosin. We're pulling the actin together toward each other. Right? This actin here and this actin are being pulled closer together. Furthermore, if we consider that within the sarcomere there are lots and lots of these myosins and lots of these actins, it's more of a three-dimensional thing here rather than just two-dimensional. We have to imagine a lot of these myosins interacting with a lot of actins, and there are lots of myosins in this three-dimensional space organized as a sarcomere, all of them pulling. Plus, we have sarcomeres lined up that are all doing this simultaneously. That should have the effect of shortening this myofibril overall. Okay? All right, now, that's how we get the contraction of this muscle is through this sliding filament mechanism. But what we haven't addressed is the calcium issue itself. We know from this figure here, when calcium levels are high, we know from this figure, high calcium leads to the cross-bridge binding, right? So we know that calcium is important in this overall mechanism, but what we need to do is talk more specifically about the calcium and what's happening at that level. Okay? All right. Now, here is our muscle cell. That's a myofibril, that's a myofibril, that's a myofibril, and that's a myofibril. So multiple myofibrils laying along linearly through this very long muscle cell. Within the muscle cell itself, I would expect mitochondria, because we need ATP, obviously, to do muscle contraction, because ATP is involved in the binding of that myosin head to the actin. Also located here are these invaginations of the plasma membrane. So here's my cell membrane around the outside, and here are these openings into the cell membrane in blue that come in and lie along each of these myofibrils. Okay? So these blue are opening to the outside. They're in pocketings through the plasma membrane down into the cell. These openings that come in, so this is cell membrane. We have these kind of tubes coming in. These are referred to as T-tubules. T for transverse. T for transverse tubules. These are T-tubules. But notice that the outside world, right, is here. That's extracellular fluid out here. Well, so's that. That's extracellular fluid, and so's this. Okay? Now, adjacent to these T-tubules is here of drawing it like this. I'm drawing it very well. It's got a good picture there. But in black, this is a modified endoplasmic reticulum called the sarcoplasmic reticulum. Right, the sarcoplasmic reticulum. The sarcoplasmic reticulum is the endoplasmic reticulum of muscle cells. And they're modified a little bit in that they're organized like this, still highly convoluted like an ER should be, but they run right up against that transverse tubule. Okay, they're right up against each other. Okay? All right, so let's make this work. All right, here we have an axon terminal. That's a synaptic terminal where that synapse occurs between the neuron and a muscle cell. Okay? So where that neuron comes in here, this is the muscle, this is the neuron. Okay? That's a synapse, but it's a very specific type of synapse. It's referred to as a neuromuscular junction, because that's where the neuron and the muscle cell are coming together, is at this neuromuscular junction. This piece of it here, the muscle piece, is called the motor end plate, because this has to be a motor neuron, because it's going to tell the effector cell, in this case the muscle cell, exactly what it needs to do: contract.